All right, so today I'm going to be working on updating methods for RNA-Seq using liminal. So I'm going to come to my notebook, start a new entry, and I'm just going to say updating methods for RNA-Seq. Okay, cool. I'm not going to mention a previous entry, but I'm going to say it's probably a little bit of background. So our lab will be doing this more in the future. And /user. I'm just going to say we got this user is currently running an analysis. Okay, cool. Okay. All right. So it's been a while since I've done an RNA-seq analysis, so I'm just going to put a little section here. I don't think. You know what? I'm going to go into Oxford Bioinformatics. I'm going to come here, content, and I'm just going to search RNA-seq. I don't know the best way to search this. All right, I guess got to verify my human again. Okay. Gonna say from 2021, because I'm pretty good at things that are older than that. Quality control for quantifications of large cohorts. I'm not particularly going to be working with large cohorts. That is something interesting that I want to have. So I'm going to say I'm just going to start, large cohorts. And this will be kind of on my reading list for later. Post sequencing summary control is a crucial component. Post sequencing quality control. Applied to cohorts, hundreds, thousands. Okay, so it's got a GitHub too. Okay, maybe I'll copy this link address, come back here. Cool. That's something that I might be interested in. All right, explore a new pipeline to explore active transpose. Well, sounds interesting, but not something I really think we're interested in. Analysis of temporal, interesting. Okay, I kind of like. Did I already copy paste this? I kind of like this too. Pipeline environment. Okay, cool. Yeah, so I think this here is kind of a good start for me to, I'm gonna do this. Now, I've got this discover knowledge button where I can, basically it'll read through my entry and see if there's anything relevant that's already happened in our lab. Or I can come here and talk to a chat bot. It's more interactive instead of it just parsing what I already have. But yeah, I like what I do to be useful to other people. So there's kind of a couple components to that. One is when I save this and I come back, everybody in my lab will be able to see, updating methods for RNA-seq, and they can come in and see what I'm doing. The other aspect is I want to train our AI, or augment it, I guess is the better word, to learn from this entry that I just did. So if somebody is saying, hey, to the chatbot, I want to do RNA-seq in the future, or I want to do RNA-seq now, it'll know that I've already done some work that might be potentially helpful to them. I like to work at very granular notebook entries. I like to open something and then close it. And so what I'm going to do is I'm going to add a conclusion. Awesome. And I'm just going to complete this. This is now complete, right? I did barely, took me hardly any time. It actually was good for me to slow down and write my thoughts. If I wanted to start a new notebook entry, I could actually add the previous entry. And I could then search by, things that are owned by me and, reference a particular entry that I could then just go back to, really quickly. That's how I'm doing my work on my own. And at the same time, I am benefiting my lab based on what I know is going to be contributed to the full lab as well.